Dear ladies and gentlemen, thank you for watching our research presentation. My name is Lutfi Adi Pradista from Magister Animal Science Program, Faculty of Agriculture, Universitas 11 Maret, Surakarta. Today, I will present our study entitled Metagenomic Analysis of Non-Pathogenic and Pathogenic Cecalibacteria Profile in While Supplemented with Betaine at the 9th International Conference on Sustainable Agriculture and Environment. Higher ambient temperature is a harmful issue in tropical islands such as Indonesia. Poultry industry have more affected, causing higher ambient temperature related to the higher stress response. Research shows that betaine is essential in increasing feed efficiency, stabilize cell pressure, and improving performance. Difference in poultry utilize with nutrients cause different conditions in the digestible tract. Different conditions such as pH, available energy, and others may change the pathogenic and non-pathogenic bacterial composition. We hypothesize that giving betaine as a feed supplement on quail has higher non-pathogenic bacteria. Research to find the different composition of pathogenic and non-pathogenic bacteria secum on quail with betaine supplementation has never have done. This research aims to see the effect of betaine supplementation on pathogenic and non-pathogenic quail secum bacteria. Metagenomic next-generation sequencing is a suitable method to see microbial profile from animals, human, or other samples. Based on 16 RNA region V3 to V4, we can see taxonomic profile from pathogenic and non-pathogenic bacterial samples. This research focused on non-pathogenic bacteria in the genus Collinsella and Corynebacteriaceae UCG002. On the other hand, the genus Olsenella, a pathogenic bacteria. This study is experimental design with completely randomized design with 3 treatment levels based on betaine supplementation. The treatments include C as the basal diet, B1 as a basal diet with 0.15% betaine supplementation, and B2 as B1 with the withdrawal of 0.15% betaine supplementation. The second sample on quail was carried out at 129 days for treatment C and B1 and 159 days for treatment B2. The sample storage of the second fluid was carried out according to the Thermo Fisher scientific protocol, where samples were stored in a 1.5 ml tube and then given in a liter solution until all samples were submerged. The collected samples were then analyzed at Novokin Company for microbiota profile. Microbial profile visualization is described using Corona. Analysis of variance and DMRT post-hoc tests were conducted to determine the significant level of the observed microbial composition between each treatment. Elsevier version 4.1.2 performed statistical data analysis according to the R-core team procedure. Based on reference, we find 3 bacteria that have an important role might be affected with given betaine supplementation in the diet. Valentin et al. State that genus Collinsella plays a role in amino acid metabolism. Frambois et al. State that the genus Corynebacteriaceae UCG002 increase SGFA concentration and act as an anti-inflammatory related to the function of betaine on poultry. Ratrianto et al. State that betaine improves nutrient efficiency and digestive cell pressure in poultry. Yan et al. State that the composition of non-pathogenic bacteria has a negative feedback on pathogenic bacteria. So in this research, we confirm the genus Olsenella as pathogenic bacteria. Corona chart from several observation samples show that the composition of the genus Collinsella and Corynebacteriaceae UCG002 tends to increase with betaine supplementation in several research samples. Betaine supplementation in the diet also show a tendency to reduce the composition of the genus Olsenella as pathogen. The withdrawal of betaine supplementation in B2 show a microbial quality degradation pattern close to the composition of the control treatment. The betaine supplementation in B1 significantly increased the relative abundance of the genus Collinsella and Corynebacteriaceae UCG002 compared to the control treatment. On the other hand, betaine supplementation or B1 treatment in the relative abundance of the genus Olsenella show a significant decrease compared to control treatment. Young et al. State that non-pathogenic bacteria have an anti-inflammatory role, so the increase in anti-inflammatory bacteria can reduce the composition of pathogenic bacteria such as the genus Olsenella in the digestive tract. Relative abundance of the genus Coriobateriaceae UCG002 show a decrease in tense due to the withdrawal of betaine supplementation. The genus Coriobateriaceae UCG002 plays a role in increasing the concentration of HGF-A in the digestive tract. Liu et al. State that HGF-A in poultry has a substrate in producing energy. HGF-A can lower the digestive tract pH concentration and inhibiting pathogenic bacteria. Consistent results were also shown by negative feedback that the composition of pathogenic bacteria of the genus Olsenella increased with the withdrawal of betaine supplementation in treatment B2. Based on this result, we conclude that betaine supplementation on lying while improved the quality of the psychomicrobial profile. The increased number of non-pathogenic bacteria confirmed the reduced pathogenic bacteria in the wild cycle. The population of Collinsella Coriobateriaceae UCG002 enhanced, whereas Olsenella decreased due to betaine supplementation. After the withdrawal of betaine supplementation, bacteria in the genus Collinsella Coriobateriaceae UCG002 and Olsenella show a return tendency to the control treatment composition. Thank you for watching and stay healthy.